Okay. All right. Thank you all for joining. I'm Katharine J. I'm your host for today's HIV Grand Rounds. I'm really excited, really pleased to have Dr. Katharine Barr here. She's an associate professor of medicine in the Division of Infectious Diseases at the University of Pennsylvania School of Medicine. She completed her internal medicine residency and achieved here at Virginia Commonwealth and then fellowship in infectious diseases at University of Alabama. Dr. Barr is a translational physician scientist who runs a molecular virology lab, which focuses on mechanisms of virus transmission, pathogenesis, and persistence of HIV and other viruses using non-human primate models as well as clinical cohorts. And she truly embodies the translation in translational science, bridging basic science and clinical trials. She holds leadership roles in the Penn CFAR and the NIH Clinical Trials Network's Advancing Clinical Therapeutics Globally, or ACTG, and the HIV Vaccine Trials Network. Including serving as director of the Penn CFAR Virus and Reservoirs Advanced Technology Core and chair of the ACTG Reservoirs Remission and Cure Transformative Science Group. She also serves on multiple scientific advisory boards for HIV vaccine development, and she's deeply involved in graduate student and trainee and early career investigator mentorship, teaching in the medical school at Penn, and mentoring an impressive number of individuals at every level of training, some of whom are right here in this room. And so I feel actually really lucky because we have this kind of great Penn, Barr Lab, UCLA pipeline going. So that's lucky for us. Dr. Barr has developed an impressive body of work elucidating adaptive responses to HIV-1 infection and characterizing in particular neutralizing antibody responses in HIV-1 infection, including autologous neutralizing antibodies, or ANABs, and viral escape from both passively administered antibodies and ANABs, some of which you will review with us today. So this work is informing the development of HIV prevention and cure strategies in this very moment. And she cares for patients. So Katie, I don't know how you do it all, but it's great to have you, and we're really excited to hear about your work.
Thank you so much, Kara. And it's so nice to be here. And I will say that I've had the opportunity to work with Jonathan back in his East Coast days. And I see Greg Whitehill over there, and I see Hunter Courtney over there. And so it's so nice to have people who, these two directly informed this work. So I'm very thankful to those who, before their UCLA times, contributed to East Coast science so that we could, so I could present this work here today. It's really lovely to be here. And I hear it's quite humid today for UCLA standards, but it's actually lovely. And so it's a wonderful, for this East Coaster, it's way better than I could have expected. Okay, so I'm going to try to talk about antibodies in HIV. And, vaccines are quite controversial right now, but if we think about them scientifically, almost all of the currently approved vaccines in the United States right now have an antibody as their correlate of protection. And so we know that antibodies are incredibly important in preventing and treating and curing viral infections. Even in some of the most challenging to treat infections like hepatitis C, where we have a RNA virus, very fast replicating, very diverse global disease, we also know that antibodies are associated with rapid clearance of that virus. And so it's also been shown, even in very challenging RNA viruses, that antibodies are very important. But it's really been completely unsuccessful, any attempt to use antibodies to functionally improve HIV disease via cure, via therapies, or at this point. And that is because HIV is probably the most challenging virus to neutralize of all of the viruses that we're studying for a bunch of reasons. If we think about neutralization in HIV as an antibody that can bind the trimer, the envelope trimer on the virus, thus preventing it from infecting the next cell, then we know HIV is highly evolved to escape neutralization. So this is actually the cartoon sort of replica from CROI, the conference, but it does a nice job of showing a sort of a representation of what a virion looks like. And I don't know, just this little, oh yeah, the arrow shows up. So in addition to a small number of functional trimers that sit on the virion, there's also a bunch of other non-functional trimers that are highly immunogenic. So there's little GP41 stumps, there's monomers or dimers that are not functional, but have all of their viral properties hanging out for the immune system to see. And the immune system sees those and says, that's a virus, let's attack it. as opposed to the very small number of actual functional trimers that sit on the virion. This is densely coated by a cell-produced glycan or sugar shield, and that is much less obvious to the immune system as being a component of a pathogen, and therefore it's far later that we get good responses against functional trimers. Of course, HIV also directly infects and kills CD4 T cells, which is the part of our cellular immunity that provides help to antibody maturation, and therefore we mess up and delay the development of more functional and relevant antibodies. And of course, HIV is extremely diverse. So within any one individual, we can have up to 10 or 15% diversity within envelope circulating in an individual. And if you take that amount of diversity and spread it across a region or the world, even if you develop a very good antibody response against the virus, one virus or one person's virus, that's oftentimes ineffective to handle the diversity around the rest of the region or globe. And so what happens is that in HIV, the antibody response is always behind. It is too little too late at every step of the infection process. So this is adapted from looking at sort of waves of antibody responses over time. But what we see is within the first few weeks of infection, we get our first de novo antibody response. These are non-neutralizing. They bind to some of those immunogens that we showed on that virus, but they don't bind the functional trimer. And therefore, we can use these to diagnose HIV infection, but they do not actually drive virus evolution, nor do they prevent infection of new cells. We don't actually get neutralizing antibodies in most individuals for about 60 to 90 days. And so by the time those first functional neutralizing antibodies have developed, the horse is out of the barn, virus has infected a long-lived cell, we have a permanent reservoir many, many weeks prior to that. But also that slow evolution allows virus to rapidly escape. And therefore, by the time you get a neutralizing antibody response, you've already escaped in terms of your virus populations. And we're going to talk about that more moving forward. Now, of course, some individuals with HIV go on to make cross-reacting or broadly reactive neutralizing antibodies. These people who make BNAbs, we think it's about 20% plus or minus people who have ongoing virus replication for 1 to 3 years. So it's a rare fraction of individuals. And in the vast majority of those cases, when you do develop a BNAb, your virus population has already escaped that. So it's not directly relevant for that individual, but those BNAbs are models for the sorts of antibodies that we'd like to make as either products of vaccines or as reagents or medicines to use on their own. So why do we think it makes any sense to even use antibodies if they're just flailing at every step in natural infection? Well, the theory of trying to use antibodies as interventions against HIV is sort of one thing to think about is if HIV is working that hard to escape antibodies, then maybe there's something to the fact that if we could fix them or make them better, they might actually be quite effective. And so what myself and many other people in the antibody world are trying to do is change that timing relationship between the development of broad and potent antibodies and the ability for the virus to escape. So for example, if we can get highly effective antibodies at key clinical junctures before the virus has escaped, we think they could prevent infection, suppress viremia, and potentially clear infected cells. So for prevention, that's using a vaccine before or a passively administered antibody before someone comes in contact with the virus. And so we think about that as having these antibodies present before exposure. But for treatment and cure, what I'm arguing we can do is take that autologous antibody response, use antiretroviral therapy to lock that virus down so it can't continue to evolve and escape, and then push the antibody response via immunotherapies or vaccines or delivery of passive BNAbs through other means to deliver a broad and potent antibody that the virus does not have the opportunity to escape from, so that when the virus comes back, you've got your good antibody ready to go. Okay, so is there any evidence that autologous neutralizing antibodies in any situation have actually shown activity in people with HIV? Well, there's actually just a couple case reports that I'm aware of. And one of them was published a couple years ago from the group at NIH led by Taeuk Chung. And what they described here is this participant 30 for whom this person had stopped their antiretroviral therapy and maintained virus suppression for years. And so what you're seeing at blue here is sampling of the antibody response, and in red, and all these triangles are viral load assays showing over these 1,200 or so days that there's no active virus replication, with a couple tiny little blips. And what they showed when they sampled the immune globulins from this individual is that the antibodies in this person very effectively neutralized the virus that they could recover from, the provirus that they could recover from this individual over all this time, until at this point in time when virus broke through. And what it turns out happened is this person was actually super infected by a genetically distinct virus that was not targeted by their range of autologous antibodies. And so this sort of shows a proof of concept how autologous antibodies can control virus, but then also a weakness. If you have a highly divergent virus that enters the system, you lose that virus control. A similar sort of picture has been described in one of the E-Clear participants. So this is a clinical trial in which BNAbs were given at the time of our initiation. And one of those individuals has maintained post-intervention control for, I think, almost 4 or more years now. And what researchers in the Silicano lab and others have shown is that the antibody response in that individual very potently targets the autologous antibody, the autologous virus. And they think that's at least part of the reason why that person has maintained such good virus control for so many years. So we also looked at autologous antibodies in a recent clinical trial that was conducted in Philadelphia through the Wistar and Penn. And basically in this clinical trial, individuals were given broadly neutralizing antibody and interferon. Their therapy was stopped. And then they were monitored to see if virus would return as the BNAbs were still at high levels, as the BNAbs waned, and then afterwards. And what we showed in that study was that most of the individuals maintained virus suppression while the BNAbs were at a very high level. Just 2 of those individuals had rebound. The majority of people rebounded as the BNAbs were waning in the system, which makes sense. But then there were 3 individuals who actually had prolonged duration of virus suppression after the BNAbs had waned from the system. And so we characterized the autologous antibody response in those individuals. And what we showed is that those 3 individuals here who had substantially higher levels of autologous antibody responses against their reservoir viruses were also the people who had marked periods of post-intervention control after the antibodies had waned. So this is another example of autologous antibodies potentially correlating with post-treatment control. Okay, so let's switch for just 1 second to the clinical side. And I think we all recognize that early antiretroviral therapy is essential for best, standard of care clinical practice. So we know people with HIV benefit immensely their personal health from starting antiretrovirals as soon as possible. It stops disease progression. It returns people to health. We also know there's a public health mission for starting antiretrovirals as soon as possible because undetectable equals untransmittable. So if people's virus is suppressed, they can no longer transmit the virus. And this is a very important, approach to public health. But we also think that early antiretrovirals might be an optimal setting for an immunotherapeutic cure strategy. And that's because people with early ART initiation have a much smaller and a much more less diverse virus population in their latent reservoir. We know that early antiretrovirals preserve cellular and humoral immune function. They don't get the exhaustion, immune exhaustion that goes along with any chronic infection, including HIV. And one of the metrics of this is that the frequency of post-treatment controllers, people who start antiretrovirals, stop, and then have their virus controlled without any antiviral, that frequency is much higher in people with early ART initiation, somewhere in the 10 to 15% in large cohorts that have been studied, compared to chronic ART initiation, where it's somewhere in the sort of 2 to 3% of people maintain virus control after treatment interruption. So we think this is a nice opportunity to intervene. But one of the things that we thought a few years ago hadn't been fully addressed, despite the fact that we've characterized antibody and virus interactions for decades now, is what happens when people start early antiretrovirals, what happens to their antibody response. So this is a project led by Greg Whitehill over there, who's now a second-year ID fellow here. But what we reacted to was The only study in the field that we saw available was from the MHRP. And of course, the MHRP has these amazing cohorts of individuals with acute and early antiretroviral therapy around the world. And they studied quite a bit about cellular responses and antibody responses. And in fact, they looked at a cohort of acute and early ART initiators, and they characterized binding antibodies, antibody functionalities like ADCC, and they looked at people who had started between stage 1 and stage 5. So this is between acute and early infection. And they characterized binding responses, which they saw. They characterized ADCC, which again, later in later sort of early ART initiation, there's good ADCC functionality of these folks' antibodies. And then they looked at tier 1 neutralization, meaning do the antibodies that these individuals have between week 0 and week 48, so the first year of antiretroviral therapy, are they able to neutralize heterologous viruses that are lab adapted and much easier to neutralize than any primary viruses that exist in actual people in the world? And they, it's a little bit messy here, but basically they showed no tier 1 activity, but they didn't look at autologous neutralizing antibodies. And so that's what we wanted to look at in the cohort that we had access to. And that cohort is a cohort from UCSF led by Sugi Lee. And they have a lovely program through UCSF and the public health department where individuals who are diagnosed with acute or early infection started on antiretrovirals that day and then enrolled into a study collecting their biospecimens over a course of years. So Sugi gave us access to samples from these individuals. First, plasma sample at the day of diagnosis when there's still plasma virus, and then they would start antiretrovirals that same day, and then following over time on antiretroviral therapy. And so we had 23 participants from 3 groups. One group was people who started within 60 days, likely, and interpreted 60 days of their likely infection date. Another group is early ART initiators, sort of between 60 and 120 days. And then we had a third group that we're going to show here that started with acute at acute time points, but had some period of virus rebound. And so what I'm showing here are the viral decay curves on antiretrovirals for these 3 cohorts. And the thing that I want to point out is that in acute ART initiation, importantly, a lot of these folks have very high viral loads. So their viral loads are in the 1 million to 10 million and higher levels. That is a lot of virus-infected cells. And we know historically, when individuals start on antiretrovirals at this time, they go through the normal multiple phases of virus decline. But because you're starting at, 10 to the 7th viral load, that means there's many infected cells. And it takes a long time before that virus is fully undetectable in the plasma. That's still an effective antiretroviral therapy, but we think about log decline of the viral load. And so what you end up with is at 6 months, a substantial number of these individuals still have detectable virus in their plasma. So there's still virus in the system. There's just no longer virus replication. And if we look down at our early ART initiators, again, we are starting closer to sort of set point viremia. So these people's immune systems have been able to control the virus a little bit. There's a substantially lower number of infected cells and virus. And therefore, within just a few sort of 1 to 3 months, more typically what we see with traditional ART initiation and chronic ART, those viruses are basically undetectable within just a small number of months. So here we're talking about our acute ART initiators and that had some period of detectable viremia, and those we're going to treat a little bit differently because they didn't have that sort of lack of virus replication that we believed happened with these the gray lines. Okay, so our approach was to sequence the plasma virus and to infer what we thought either in our acute individuals was the transmitted founder virus, or in our early individuals, just a consensus sequence. And so in the acute sampling, what we can see here, is that here is a representation of a very low diversity lineage of viruses. So in phylogenetic trees, genetic distance is shown by horizontal distance. And so what we're showing here is that these chunk of viruses here, these are all identical. And then down here, we have a very small number of unrelated changes. And so here we could infer the identity of the likely virus that established infection not that many days prior. And we can use that to assess antibody responses moving forward. This is another very early sampled individuals, but what we see is three lineages that are low diversity in and of themselves, but you can see these long horizontal distances between these lineages suggest three distinct viruses establishing infection, and they haven't had a chance to diversify much, but they are distinct from each other. So this is what acute sampling of a multi-virus transmission or multiple variant transmission looks like. In our early sampling, these viruses have had a time to replicate, and they started to respond to various either selective pressures or whatnot. And so you see more diversity within the virus population. So here in 8048, we're looking at a single virus transmission that reflects some genetic diversity. And in the sort of literature understanding how virus evolves in envelope, these shared mutations, like here, there's this line here that connects all of these. This means there's a shared mutation here. Those shared mutations are almost always due to escape from autologous antibody responses when you're sequencing OM. And so this is sequence evidence suggesting escape from an early antibody response in this population. This is an early sampled individual that had multiple virus transmission. This is basically what these three lineages look like after you give them time to recombine. And if you have multiply infected cells, those viruses basically combine with each other, and you end up with a very diverse virus population very quickly. And so this is essentially what chronic HIV looks like if you sample the plasma virus, but it happens much faster with multiple virus transmission in this sort of setting. And so, so in these early ART initiators, we can't infer back to what established infection, but we can pick consensus viruses that are hopefully sort of back in the tree suggesting they may be origins, and we can sample those, and we know that they were replicating at those times, but they probably are not what was initially establishing infection. So then we went on to do neutralizing antibody responses and ask if we take the plasma at week 0, week 12, week 24, after antiretrovirals are started, do those antibodies, are they able to recognize and neutralize the viruses that were present at the time of ART initiation? And so again, we're looking at the same viruses, but the antibodies are differing over time. And so at week 0, we do not expect to have any neutralization because in our acute participants, we think that they haven't started to make an antibody response yet. And so it's not going to be neutralized. In our early ART initiators, we think they've made a response, but our sequences tell us they've likely escaped from that response. And so the antibodies that are present are not able to neutralize contemporaneous virus, they targeted a prior virus. And so like we expected, there's no neutralization present at the day that we sampled that first sort of clinic visit. So our question is, over that year of 6 months to a year of antiretroviral therapy, do these participants' antibodies evolve to be able to then recognize the virus that was previously there? And what we showed is that over time, those early ART initiators did in fact grow to have the ability for their antibodies to recognize that previously seen virus, whereas those gray dots, those acute ART initiators, were not able to improve on their antibody response to recognize that previous virus. And in the red participants, those were acute ART initiators that had a little bit of virus rebound. 2 out of the 3 of those individuals were able to mount a response, at least a low-level response, to target their initial virus. Okay, so speaking of those acute ART initiators with rebound, here we're looking at the time point that virus was identified, and then the fact that shortly thereafter or at the same time is when we start to see those responses. So very quickly after virus rebound, those responses were generated, suggesting maybe those responses were primed but had not been able to make a response without that new virus replication. Okay, so this is the early ART initiators over time, and what you can see, I hope, is that in addition to developing antibody responses, they increase in potency over time, suggesting that antibody responses continuing to evolve and mature over this time that we were able to sample them. And I think that's shown very nicely with our multiple virus transmissions, because you can see not only does the antibody response better target those initial viruses, but it actually broadens to be able to neutralize more diverse variants. So again, That virus we're sampling at the time of our initiation, that is not the transmitted founder virus, that's an escape variant. And so we're watching the antibody response grow to be able to sample that first escape variant. And here is a multiple virus transmission where we see it's able to capture, to target sort of one lineage of viruses here shown in yellow. And then and that sort of continues for the first year where we only have neutralization of the yellow virus, none of the green. But then as we sample at 80 weeks or so, we see that the antibody response has evolved in breadth and potency to be able to target multiple variants within this virus population, not just that one. So we see that it's continuing to evolve and mature like that. Here's another multiple virus transmission with a similar but perhaps less dramatic. At 24 weeks, we have only one of the two variants. At 42 weeks, we're able to neutralize with greater potency both of these variants. And so to sum up this first part, we found that acute ART initiation, those who started antiretrovirals before 60 days of infection, they did not actually establish a neutralizing antibody response. There was no evidence of escape in those early virus sequences and no subsequent development of an antibody response. The thing I want to point back to is if you looked at those virus decays, those people have virus in the system for 6 months. So while these antibody responses are not developing, there is antigen, there is virus, but there's not virus replication. So it suggests an interesting sort of dynamic there. You don't just need virus, you need specific properties of virus replication. And then in those individuals that had a little bit of rebound, you can elicit those responses. And what we hope is that we can get that same response from a little bit of rebound, perhaps with an immunization or another effective sort of showing of the immune system, these viruses. Early ART initiators, on the other hand, we believe that they had already established a response that we see in terms of sequence evidence of escape, and it continues to mature in breadth and potency over that period of time on antiretroviral therapy. And again, it broadens from attacking that TF, that initial virus, to that first escape variant that we sampled at the time of our initiation. And then in multiple virus transmission, we see evidence of being able to target minor variants as well. And it does seem to increase in potency in the individuals we had samples for over years. And so this suggests a threshold effect in that you need virus replication, not just the presence of virus over time in order to initiate that neutralizing antibody response. And you can develop binding antibodies, tier 1 antibodies, but you really need that neutralizing antibody to be first developed in order for it to continue. And there's a lot of very interesting reasons as to why that may happen, and we're trying to dig into those, but we don't as yet have an answer for that. So one of the things that this study made us think about is it provides optimism for the fact that we might be able to further boost that autologous antibody response that is growing in breadth and potency on antiretroviral therapy. Maybe with a vaccination, we can make it grow a little bit more, and an autologous antibody boosting vaccine could actually have potential. And that's partially because acute and early ART initiators, we know they have these small and genetically narrow virus reservoirs. They have better immune responses, and we can boost those responses that already exist. And so we're hoping to think about boosting those existing responses as opposed to a lot of the antibody-based vaccines that are out there right now, which are looking to target naive B cell lineages that are thought to be good germline targeting, good BNAb lineages, good B cell lineages that have the potential to evolve into broadly neutralizing antibodies. This is a different approach where we're just trying to build a little bit on the responses that already exist. So hopefully we can get to be broad and potent enough for the autologous virus population, not for the entire world. There's a ton of amazing vaccine technology out there, and we're really lucky at Penn to have a lot of great people who access all of these various immunogen design and mRNA and DNA and all these sorts of wonderful approaches. And so we are working with these folks in order to try to develop this. I'm going to talk more about that. But we envision a personalized autologous antibody boosting vaccine that could be based on each individual's virus swarm, characterized just like these individuals were as they come into clinic and start antiretrovirals. You just need that one sampling in order to be able to model what is going on with the virus population, and we can design from there. Okay, so I just want to shift briefly and talk about our work in the non-human primate model. One of the things my lab does is try to go back and forth between human cohorts and non-human primates, because you can learn amazing things in human cohorts. Then you can simplify experiments, or you can make them more complicated, or isolate specific parts in the non-human primate model, go back to humans, back to the non-human primates, etc. And so I just want to show a little bit about our work in non-human primate experiments and hopefully convince you very quickly that in terms of virus-host interactions and antibody development, the non-human primate model, when infected with SHIVs encoding primary virus envelopes, they do a really nice job of seeing the same sort of thing that we see in humans. So this is a study that we did a few years ago with 18 animals infected with a SHIV encoding a clade D envelope, in which we treated them with bNAbs and antiretroviral therapy, and then looked at how that worked. So this is the first 4 months of infection. We infected these animals. You can see viral load set points in the, gosh, it's hard for me to read all those zeros, but somewhere around 10,000 is about the set point viremia. Then we suppressed these animals with antiretroviral therapy. We tried to model this after some of the first ATI bNAb studies. So we gave VRCL 7523RLS, which is a pretty potent CD4 binding site antibody. We saw a modest delay in rebound in the blue versus the red line. We suppressed again with ART, and then we did another treatment interruption. But what I hope to show here is this looks very similar to what we would see in an acute cohort of humans who were suppressed with antiretrovirals given a modestly potent antibody. And so we went on to characterize the autologous antibody responses in these animals. And what I hope to very quickly convince you of is if we sequence the viruses from these animals, this is just one animal as a representation. But if we have the transmitted founder virus, which we know in the animal model, because we know what we used in the inoculum, although we don't know that in humans. And then we look at virus from day 120, which is sort of month 4 or around the time that an early ART initiator would come to clinic. And then we look at what virus ultimately rebounded. We again see that there's nice virus evolution in our red sequences suggesting 4 months of virus diversification. And when people the animals undergo a treatment interruption, the virus that rebounds is very much associated with that most evolved virus, which is actually what we see in human clinical trials. So here we're looking at this particular animal's time course from the day antiretrovirals were started at 120. We're looking in black at the autologous antibody response against the transmitted founder virus. In blue is the autologous antibody response against what ultimately rebounds. And in red is the viral load. So again, when you start on antiretrovirals, the virus is suppressed. We can see at the time of ART, there is actually a response against the TF, which is what we inferred happened with early ART initiators, but we couldn't measure because we didn't actually know what the transmitted founder virus is. And over time on antiretrovirals, that response actually is stable and then increases slowly over time, similar to what we saw in our human cohort. And then we do a treatment interruption. Virus comes back pretty quickly. With viral rebound, there's a massive boost in the response against the TF. And we start to see that the rebounding virus, that virus that rebounds, it's actually totally resistant to pre-existing autologous antibodies, but then we see a boosting of the response as that virus returns. And so this is the same sort of viral antibody dynamics we see in human, where we see development of antibodies by the time ART initiates, and then we see those response increase over ART. And then we see the rebound virus comes from an ANAB resistant pool. And then only after rebound do we start to see those responses substantially increase, both to the sort of antigenically imprinted transmitted founder, but also this new rebound variant. And so with this, we are hoping to use this non-human primate system to be able to test some of the things that we have learned from the human cohort. So this is just a very busy slide that we stole from Mike Hogarty and George Shaw, who have done a ton of work looking at virus infection in SHIVs and immunization in SHIVs, in non-human primates. And what they basically showed is if you compare viral loads versus viral loads after vaccination, and you look at the antibody responses generated after infection versus after immunization with OMS that are the same as the infecting virus, you can see very similar responses in these animals with vaccination and infection. And so this also supports our ability to want to use vaccination to try to push these responses. And so what we envision and what we recently got funded, thank you NIH very much, is this particular study that we are just starting right now. And the goal here is to test an individualized autologous antibody boosting vaccine versus what is currently one of the best off-the-shelf germline targeting sort of bNAb approaches for vaccination. And so in our first arm here, all of our animals are going to be infected with a SHIV that encodes a transmitted founder clade D virus. They're going to have 4 months of virus replication. They're all going to start on antiretrovirals. Our arm C will be ART alone, so we'll see how much those antibody responses improve just with antiretroviral therapy. Our major arm will be arm A, where they're going to be able to have virus replication for 6 months and sort of let those antibodies brew for a little bit. And then we're going to immunize them with 2 immunogens that have OMs that are based on the viruses sequenced from their plasma at the time of our initiation. So we're going to have one OM that is sort of the consensus of those sequences and one OM that's meant to push to encapsulate more of the diversity present at that time. And we're going to compare that to a germline targeting approach, which is GT 1.1 plus BG505, which is currently being tested in some human clinical trials right now, is for therapeutic vaccine to see if it can engender either de novo or boosted antibody responses. So we're very excited about this study, which we're just getting started. But we also recently were able to test some of these same concepts within human cohorts. So there was an iCure grant that it's a UM1 consortia that was recently funded. This is led by Luis Montaner with Drew Weissman and Bob Silicano. And we have a project with basically the same group that we're doing the non-human primate study, but we're doing this within human cohorts. And so the goal is to take individuals who enroll after acute and early ART initiation. We're going to sequence their virus from their PBMCs and from their plasma. We're going to infer the virus diversity from this, generate immunogens. We have a lovely computational program that assesses the diversity and then figures out the right envelopes to encompass that diversity, as well as thinking about the specific glycan holes of those various OMs. So we're going to generate mRNA, lipid nanoparticle, stabilized trimers and immunize. We're going to do this immunization in rabbits and determine what sort of antibodies we're able to induce with these specific immunogens. And then we're going to go back to the cells from these individuals and see if in the context of like a virus outgrowth assay, the rabbit-induced antibodies will be able to prevent virus reactivation in an in vitro situation. So that study is going on in parallel. And the hope is after that non-human primate and this study are done, we'll have enough sort of data and energy and momentum to be able to try to do a clinical trial in humans with these immunogens to see whether an ANAB boosting vaccine approach would be able to lead to better virus control. Okay, so the very last thing I wanted to talk about is related to this project, and this is something that Hunter and Greg and I are trying to push through as the second part of this sequencing. So this is going back to that same non-human primate study. And what I'm showing here is the individual viral kinetics of one of those non-human primates over time. So we have virus inoculation, peak viremia. We settle down into sort of set point. They have their first ART treatment. They have viral rebound. Then second ART treatment, again, viral rebound. So we sampled these plasma viruses and PBMC viruses throughout this longitudinal study. And I'm showing them here on the right. This is the phylogenetic tree of all these different sequences. This is the highlighter plot showing the specific mutations associated with each one of these viruses. And what I want to highlight here is that if we think about compared to our transmitted founder virus, when we have this first rebound here in this cyan color, this first rebound is actually quite genetically distinct. It is the most evolved, the most sort of escaped, and we know phenotypically these are also ANAB escape variants. So these viruses is what rebounds, and we can follow that over this first rebound, and these darker blue viruses are what are circulating or replicating when we start that second ART treatment. And then we can look what happens at the next rebound. The next rebound, we see the most genetically divergent variants. Again, the things that were circulating right here at the last time point before we started antiretrovirals. That's what rebounds in our second ATI. So it's all of these more divergent variants that are key to what is able to rebound, what keeps driving infection in this model. But if we sample the reservoir, so here in yellow, we're sampling the reservoir right before that first treatment interruption, and in green, we're sampling the reservoir right before the second. What we see, in fact, is it's very, very similar. The majority of these viruses are very similar to the transmitted founder virus. And that reflects a couple of things. First of all, we are deeply undersampling the reservoir. So there's huge limitations to what we're sampling like 10 or 15 sequences over a heterogeneous pool of viruses. But across all these animals, you do get a sense of what the majority reservoir is. And in acute ART initiation, the majority reservoir is the transmitted founder virus. And that's because there's only a limited amount of time that you had virus-infected cells. Your peak virus is, 5 million. At the time of ART initiation here, where your virus replicating is that escape variant, you're 2 to 3 logs lower. So there's literally like 1,000-fold greater infected cells that could be a part of your reservoir from primary infection compared to your escape variant that is only a minor portion of the reservoir. And so that all makes sense, like stochastically, and the reservoir sort of informs what was likely to be able to be seeded into the reservoir here. But the majority of the reservoir doesn't really matter. What matters is what is most fit to reactivate. And in this case, it's these more divergent viruses. And so again, the reservoir is highly enriched for the TF, but we know that there's this selection process where you just need a minor variant. If it's more fit, it's going to be more likely to replicate. And so we wanted to know if the same phenomenon was seen in our human cohort. And so that's what we tried to do. So this is one of those pictures that we showed before of one of the early ART initiators with a single virus transmission. This is 8048. We picked these two. I'm going to blame Greg because he picked them. Greg picked these two viruses as likely representative of consensus viruses, and they were good choices. We knew they're not TFs, but we thought they likely represented reasonable viruses. So if we went back, in addition to this plasma virus, add provirus OMV that was sampled from the 6-month time point of the reservoir that we had samples. And I think Hunter did a lot of this, but Greg may have as well. But if we sample those PBMC OMVs and we add them to the phylogeny, all of a sudden we have more directionality and we can actually infer back virus evolution from transmission to what we ultimately see in the plasma at the time of our initiation. So in blue are our PBMC OMV sequences, and red are these same circles here that we're now showing in red. And what you can see is our reservoir after early ART initiation is highly enriched. It's super biased for these identical or nearly identical viruses, which we are going to infer were the likely transmitted founder virus from this individual. And at the time of our initiation, we have some evolution, and these red viruses are more like what was actually circulating at the time in the plasma. And so I'm just orienting you. So this C4 here, this is this variant. This orange guy here is down here in F2. And what we can do is also look at the antibody pressure to add to our understanding of the directionality. So if we look at week 12 plasma, the week 12 plasma is actually able to neutralize C4, where it is not able to neutralize these other variants. And if we look at the sequences and the amino acids of these actual viruses, we can tease out what is specifically going on here. So our TF here, which is represented by this virus, is neutralized. And then these various mutations in V1, these changes in the glycan shield, and here these various single nucleotide polymorphisms, as well as glycan shifts, these lead to escape of this response from that early antibody, and that makes a lot of sense. If we look at the week 24 plasma, in fact, all of these viruses are now neutralized because that antibody response has evolved to be able to sort of handle this shift in V1. So it now is able to neutralize a more broad range of variable loop 1 envelopes. And so that's a nice way of showing how these PBMC viruses, just like in the non-human primate model, are able to inform directionality of virus evolution. And as we're thinking about how are we going to make an antibody-based vaccine, this amount of information helps us think about where is this virus going, what is most likely to reactivate, how do we think about the envelopes that we want to use as immunogens in order to have our most effective response. So here's that multiple virus transmission where we had two viruses that we sort of separated in this tree. It's hard to know directionality. These had different responses in ANABs over time. But if we add PBMC sequences, the directionality becomes much more clear. So in fact, what we have is probably one lineage here, up here on top, where we have a bunch of identical viruses and then sort of nice, it looks a little bit like star-like phylogeny, but not perfect. We have another situation down here where, again, we have sort of evolution of viruses over time. And it's possible that we actually have another identified transmitted founder virus, or that this whole lineage here together is one. It's hard to say. But anyway, here's where our viruses stack up. Before, it was hard to know how they related. Here, we think it probably is this lineage. These are a bunch of recombinants pulled back, and then one or two other lineages. Now, the neutralization is a little bit more complicated here because there's a lot going on, but it just generally support this sort of new directionality. And so again, combining these PBMC and plasma viruses with the antibody response as it evolves allows us to really be able to map early virus and host interactions in these individuals in a way that we aren't otherwise. And I think that crosstalk between the two systems has helped us sort of have more approaches and more understanding of how this early process works. So in summary, the initial autologous antibody response is very much driven by systemic virus replication. And before antiretrovirals, that is always outpaced by virus escape. So we know that virus replication is needed to start the antibody response. But if we have virus replication, the virus always wins. And so there's that sort of tension there. But the way we might be able to handle that is by early ART initiation, which stops virus replication. But in theory, the antibody response can continue to progress a little bit on its own. But maybe this is a great sort of baseline for us to push further with all the new approaches to vaccination. And that's what we are hoping to be able to test in these various non-human primate and human studies. And hopefully we'll be able to have this be one component of a multifaceted cure strategy that could lead us to ART-free virus control. I think that in my, what the sort of thesis of our lab is this non-human primate model of HIV persistence does show a lot of parallel virus-host interactions, and therefore we can use it to have crosstalk between the two systems. We can inform each other in terms of experiments to conduct in the non-human primate system, in terms of new approaches to think about with humans. And so hopefully that's sort of the driving force of what my lab is trying to do in the next few years in that regard. So thank you so much for your attention. I should highlight very much Greg and Hunter, who did a ton of work on these projects. Thank you so much. Also Ryan Krause, who worked very closely with him, who just started his own, just left the lab to go to graduate school as well. So I guess I only tried to sabotage he and Hunter a little bit. It was totally ineffective, and they've carried on with their careers. So that's wonderful. It's a very collaborative approach. We do a lot of work. All of the human work is super based on very generous study participants, and we are also very dependent on funding, which we hope will continue. So thank you very much.
Thank you, Dr. Barr, so much. That was terrific. I love your talks. It's so exciting to see what's coming in the future from my perspective as a clinician and a clinical investigator, and the clinical trials that could be. So we'll take questions if anyone has any. And then for those online, please do drop your questions in the chat, and then we'll read them out loud because unfortunately we can't hear you if you speak. So any questions in the room? Yes. Please identify yourself.
So, whether the cell might be virus or the virus infecting with CD4.
You can repeat the question.
Yeah, so the question was whether autologous antibodies bind with free virus, or are you asking about virus infected cells or virus that has bound to CD4 already? Yeah, so most broadly neutralizing antibodies are designed to, well, autologous antibodies may bind to all forms of envelope in various stages, and we don't fully know. A lot of the antibodies we're aiming for are looking for that stage, like for the envelope before it's been triggered by CD4. There's a lot of exciting work by people like Andres Finzi looking at trying to induce CD4 engagement, open up that envelope, and then a lot of otherwise non-neutralizing antibodies, which there are a ton of non-neutralizing autologous antibodies that are actually able to bind to envelopes that have engaged with CD4 or otherwise sort of in that different energy or conformation state. And so then you make a whole bunch of previously impotent antibodies very able to sort of bind to either virus infected cells or to neutralize free virus. So most of our neutralizing antibodies deal with pre-CD4 engaged virus, but there are a ton of non-neutralizing antibodies that are also able to handle that CD4 engaged also.
Maybe so they can open the primer so that antibody, which neutralizing antibody can bind and.
I don't think we've been able to demonstrate. No, I don't think that happens. I don't think non-neutralizing antibodies are able to trigger envelope. There are, like, soluble CD4, other things can trigger envelope. And that in theory makes it vulnerable to non-neutralizing antibodies becoming neutralizing. But I don't think that that's a phenomenon that's seen with neutralizing antibodies.
Doesn't they can open up the hyperconformation for SARS-CoV-2. Now the neutralizing antibody can bind like the virus. So I'm just wondering whether this, say during HIV.
Yeah, I do not think it is the same mechanism of sort of perhaps. There's a lot of interest in using CD4-like triggers, but I don't think that those are the circulating non-neutralizing antibodies in HIV. Of course, there are subtle differences between SARS-CoV-2 and HIV that are why we were able to quickly generate a neutralizing antibody in SARS-CoV-2, and 35 years later, we haven't for HIV.
Okay, John.
Yeah, that's a good question. So the question is, what is known in elite controllers about their autologous antibody or broadly neutralizing antibody responses? The majority of elite controllers whom have been characterized, their control is mediated via potent cellular responses. That's what we think. GWAS studies have identified certain HLA types, which are associated with good cellular responses against conserved GAG regions as being a key determinant of elite control. There are rare reports of elite controllers who probably have lower level viremia as opposed to no viremia. Some of those individuals have effective autologous responses against their relatively narrow virus pool, but I think those are the minority and not the majority. There are an increasing sort of body of literature in post-treatment controllers. And again, the mechanisms of elite control, which is people who control primary infection, versus post-treatment control, which is people who control virus after a period of antiretroviral therapy. Those mechanisms are really different, but there's an increasing body of knowledge about post-treatment controllers. And some of those individuals, which we think largely it's sort of NK cell properties that's associated most specifically with that group, but there are some good sort of broad or effective autologous antibody responses in post-treatment controllers as well. So that's an evolving area of study that I think is rich for new discovery.
I'm going to take just one question online, which is kind of a broad question, so I'm going to reframe it slightly. And it's really about, we talk about the reservoir, but how do we measure it? how do we measure the relevant reservoir? And maybe that's a big topic. But especially in thinking about like
Yeah.
what you're saying right here, which is right, like the rebound, the reservoir, the virus that's going to rebound is
Yeah.
not necessarily the virus that makes up the predominantly the.
Yeah, I think it's a great question. There's a constant evolution in how we think the relevant reservoir should be studied. For a long time, we were looking at any cell that had any provirus in it, and that leads us to the 99+% of the reservoir that has defective virus, so that virus can never reactivate. So I think we've moved as a field to think about intact or replication competent virus. But we know only a small fraction of that, and I think this talk tried to show that, is actually what we call rebound competent. Not only is it a virus that can reactivate in a well, but it's also a virus that can reactivate in that host's immune environment. So it has to be able to escape dominant cellular responses, dominant autologous antibody responses. And we think mostly about the autologous antibody response as a driver of the rebound competent virus because it's easier to study. And so like the Silicanos and John Lee and myself, we've all characterized the requirement of viruses to be autologous antibody resistant in order to reactivate. But there's likely other immune pressures that are not as simple or straightforward to study that are also very dominant. So how do we measure that reservoir? It's very challenging. You have to both characterize the cell, you have to characterize whether that virus is intact, whether it's in a part of the genome that's likely to be, not deeply latent and reactivatable. You have to think about that virus and whether it's able to escape dominant immune pressures. And so I think that is, a $50,000 proposition to characterize each of, any one individual's reservoir for what is most relevant. And so that's one of the reasons why it's so very challenging.
All right, and John.
I was going to ask a question, but I think you've already answered it. That answer. I was just asking in terms of replication competency of HIV and how that complicates the autologous antibody vaccination strategy and analysis.
Yeah. So, sorry. The question was, how does replication competence or incompetence of the reservoir complicate evaluating the reservoir for how we're going to design an immunogen? And I will say that if you look at the intact viruses, they are much more likely statistically to also have a fully intact virus, but it's not perfect. So you probably, like the intact reservoir probably is, 95% correlated with the genetically, like entire virus intact virus. But if you're talking about, it's still a big divergence. So it's very possible that when we look at our intact viruses, we are also including some defective or replication incompetent viruses. But probably we get the general gist of the types of viruses that the type of OMs that are informing sort of like where our antibody and virus interactions have gone. So we sequence OMV because it's more straightforward and we're not looking at a fully intact virus because we think it's likely if that OMV is sampled multiple times, it's likely to be part of some replication competent virus that's like fully intact, but we're probably missing, it's not a perfect correlate.
Lots of challenges. You're doing a lot to address. Any other questions? All right, well, thank you so much.
Thank you very much.